All right, so let's get down here to some transcription. Where are we at? Where are we at? There we go. All right, so the I can statements are, I can explain the process of transcription. So first of all, let's define that process. Transcription is simply converting the code of DNA, A's, T's, C's, and G's, into the code of RNA, A's, U's, C's, and G's. Pretty much the same language, and that's what it means to transcribe. It means to convert something in one language to something else in the same language. For example, doctors will audio record their meetings with their patients. You may hear doctors after they leave you talking into a microphone or to a computer about your visit. Somebody else listens to that recording and types it up. So same language, but they're just converting it from verbal to written. Here we're staying in the same language, we're just converting it from DNA to RNA. And so that's why it's called transcription. And it is one step in protein synthesis. It is also the final step in RNA synthesis. So when we are producing RNAs like tRNA and rRNA and small nuclear RNA and SRP RNAs and RNAis, all of those RNAs, this is it. Transcription is it. We make the RNA from the DNA, and then the RNA goes and does a job. It doesn't go to translation. So that's the process of transcription. It occurs when RNA polymerase binds to, it has 3 steps. RNA polymerase binds to a region on the DNA, which we'll talk about in a second. It then runs down the DNA, reading it backwards. So it's going to read the DNA 3' to 5'. And let me draw a little picture of this so you can see. Oh, there it is. It's like, where's my thing? So if we are building, let's say, an mRNA molecule, 5' to 3', because remember you can only add to the 3' end, so you have to build in the 3' direction. And if DNA is going to be anti-parallel to that, then the DNA has to be read 3' to 5'. So let's say this is the gene, it'll start reading it here, it'll read down and stop at the end. Okay, so this is the only time you're ever going to hear 3' to 5' is the reading of DNA. Anytime we build DNA, 5' to 3'. Build RNA, 5' to 3'. Even when we read RNA on the ribosome, the mRNA gets read, the ribosome, it reads it 5' to 3'. So literally everything is 5 to 3 except reading that DNA. So anyway, as it reads that DNA, if there's an A here, it's going to bring in a U. If there's a G here, it's going to bring in a C. If there's a T here, it's going to bring in an A. Okay, so it's just going to, RNA polymerase is going to build these, or bring in these bases to build this molecule. So that's called elongation. So we elongate the RNA, we add the bases, and then we get to the end and we terminate, and it all kind of falls apart. So that's the process of transcription. The role of RNA polymerase is RNA polymerase does it. You know, in DNA replication, we had helicase, and we had, gosh, I don't even remember what all, topoisomerase and ligase and DNA polymerase 1 and DNA polymerase 3 and primase, all these things. Here we have one. We have RNA polymerase, and that's it. Now, some students get those confused. They're like, I don't know when we make when we use DNA polymerase and when we use RNA polymerase. Here's the deal. If you are making DNA, so you're going to read the DNA to make new DNA, that's going to be DNA polymerase, because think about what the word means. ACE tells you it's an enzyme that makes a DNA polymer. If you're going from DNA to RNA, through the process of transcription, then you're using RNA polymerase, because again, it's an enzyme that makes an RNA polymer. Okay, so that's an easy way to remember which one's which. So that's the job of RNA polymerase. It gets in there just like helicase does in DNA, breaks it apart. It does the job of helicase, DNA polymerase 3, pretty much all of the enzymes that are needed for transcription that would be different in DNA replication, RNA polymerase does all of those jobs. So it's a pretty amazing enzyme. I can explain the role of the promoter. The promoter sounds like it should be something big. It is actually a chunk of DNA. So if the blue is my gene, let's say this is insulin. Maybe I need some insulin, and so I need to transcribe this gene so I can send the RNA to ribosome to make some insulin. Right above, so here's going to be the first base of that gene. Above that is this section of DNA that's not a part of the gene. It's not a part of the coding, I should say, of the gene. It is a part of the gene, but it doesn't get coded. And it is the promoter region. And basically what it is it's a landing site that says, hey, RNA polymerase, right here, this is where you bind. That's what it does. And so in prokaryotes, that's all that happens. The RNA polymerase binds to the promoter and it zoop down and transcribes the transcription unit, gene. In eukaryotes, of course, we've got to be a little more difficult. But it's good, though, because what it does is that when you have multiple steps that have to happen, it prevents it from happening accidentally. So we don't want to waste energy making proteins or RNAs that we don't need. And so we have a couple of extra steps that are added, or an extra step, really, that we talk about here that's added to kind of just make sure it doesn't happen accidentally. So before the RNA polymerase can bind to that promoter, in eukaryotes, like you and other animals and plants and fungus and protists, first these transcription factors, real creative name there, transcription factors have to bind to the TATA box, which is just a TATA. It's just a series of bases. Those transcription factors land there, and once they bind there, then the RNA polymerase can bind. So the RNA polymerase can't bind to an empty DNA. It has to have those transcription factors first, and then the RNA polymerase can bind and break open that DNA and start transcribing it. So that's the role of the promoter. That's just the landing site for the RNA polymerase, the attachment site. The role of the terminator, of course, means to end. It is the end of the gene. It is a section of the gene. It's just a sequence of DNA, and it's not necessarily always the same, but it tells RNA polymerase where to detach. It says, hey, this is the end of the gene. This is all we need. You can stop. And so RNA polymerase stops doing transcription. It stops bringing in new bases. It detaches. It releases the mRNA, and then the DNA, it helps the DNA get wound back up and bound back together. So the terminator is just a segment of DNA that tells the RNA polymerase to stop. And then the role of the TATA box, we've already talked about that. The TATA box is where those transcription factors bind. It's just an extra step in eukaryote initiation of transcription. And then the last I can statement says, I can explain the role of transcription factors. They are the things that have to bind to that TATA box. So in us, transcription factors come in, they bind to the TATA box. RNA polymerase comes in, binds to the promoter, and then zoop, right down. So that's how it happens in us. So that's transcription overview. I hope that helps. If you've got questions, let me know.